There we go. All right, so I was going to make, obviously, a copy of this just in case for those that miss it or absent or sleeping in or whatever refers to it. I'm just going to go over Cnidarian, which is the second invert phylum. There's a little short video today, very short, not a lot of high demand, but I want to reiterate, and I'll probably put an entry here. I was typing it, but the internet kept going in and out, and it wasn't something you needed right this instant, so just reminding you of kind of the schedule this week's one. We'll have a quiz that I'll open up. It'll just be over, and I'll actually show you on the slides, those that are here, plus it'll be on the video, a reminder, because I'll get 15 emails going, is it over the entire PowerPoint? Have we went over the entire PowerPoint? No. Is it over this? So I will take 13 seconds at the end of this short presentation on Cnidarian, just one phylum, it's not as long as last week's. And then go from there. So I'll make that kind of clear with that. But it'll go straight from, I basically, as you know, I take the PowerPoints and I just turn them into quizzes. So I will make that. And then reiterating to last week, very good on those that came up and got a lot of work done on your project. Had a lot of people come up and take advantage of that. Do not procrastinate. I know that's the number one haiku type, if that's used right, that we're doing a lot of right now. So that's why I didn't want to give too much on this first part of the week. Like I said, the quiz, short quiz, which is just over the stuff lectured over last week. And hopefully that you'll be able to put in some time of coming up here. If you haven't, you need to do that, okay? I don't want to have any of these excuses why, you know, in these big Gettysburg Address articulate emails about why for 3 weeks you couldn't get out of bed or set your schedule up, which I'm fully open to come up here and work on this, because no, okay? So make a notes for that. As many of you, like I said, a lot of you have already finished it. Great, so anyway, so I'll go ahead and get started here. I'll turn the light off here. We're going to just talk about Cnidarians. It's just one of the phylums, and then we'll be done with that part, and I'll conclude everything up. I'll go back over, like I said, to point out for those that are taking side notes or whatnot about what exactly the quiz will be over, that I will post on Thursday, okay? So you got a few days, obviously, for that, and it's just over a handful of slides. All right, I'm going to flip off this light so I got better lighting. Okay. Oh, wrong one. All right, so Cnidarians, that's pronounced with the C there, okay? This is one of the few words that I actually can pronounce. Get that in the center there a little bit there more. Okay. All right, so basically the Cnidarians, as we talked last week, was sponges, okay? And the whole simple point of that was just showing you the first bottom rung of the animal kingdom, okay? So now we're in Cnidarians, which basically are made up of all your hydras, which look like upside down little trees. I got pictures of them. Jellyfish, hopefully we're all familiar with a jellyfish. There's so many different types. Sea anemones, which are like a hydra in some cases, but they're kind of a little more squatty body, but they got a lot of tentacles. And then corals, like coral reefs, okay? A lot of people don't a lot of times associate coral as a separate, simple organism, but it really isn't very important. Okay, so I put the bullets here nice and easy, talk about the specialized cells and so on they have, and I made sure and underline them so they would jump out at you so you wouldn't overlook them when you're studying that stuff or however it is you're doing this, okay? They're basically soft body, okay? So if you ever picked up a jellyfish down at Sanibel or something, they have those tubularian ones, the most common ones that you could come up, and as soon as you picked them up out of the water, they just kind of like a big slime ball just kind of covered around your hand. Or if you had one stuck to your leg, drifted up to your leg, and maybe little syndocytes stung you, it's very possible. I mean, not always. I had a long sleeve shirt on, so I don't have to worry about the little things getting me, okay? But basically, the Rhamnus central mouse. So this is one of the few phylums that has a radial symmetry, like we talked about last week, bilateral, left and right. That's what most vertebrate animal kingdom groups are, but there are still a few phylums that have the piece of pizza type central disc radial symmetry, okay? All right, so basically they get their name from a thing called, that's pronounced cynocytes or nosatocysts. I mean, you can kind of say it, the C can be silent, it doesn't matter, okay? But basically this translates to the term stinging cells. So if you've ever been stung by a jellyfish, that's what's getting you, right? And I'll show you a little excerpt, little picture of kind of what they look like. But they're located in little, basically specialized cells that are in tuned to the environment, that are up and down the tentacles on something type of an cnidarian, right? These cells are used basically for defense and capturing prey. So it's almost like an involuntary reaction. It's almost like a flinch, okay? So in some cases, depending on the level of the situation, whatever, like I said, you could delicately, kind of like handling a lot of, like it always blows me out, people handle very dangerous snakes, and you always think snakes are immediately going to bite you and so on, but you see them, handling them and, calm, and just like any animal, they get scared, they lash out. Well, they're no different, okay? So a great example is in the comical movie, or in the Disney movie Finding Nemo, okay? The part where, after he's sending Nemo off to school, he tells him they live in a sea anemone, which is, all the tentacles, and they tell, Marlin, his dad, tells Nemo to rub up and down on the tentacles, and you see him over there being silly. Basically, all that's doing is familiarizing, just like a voice with an animal or something like that. It's letting that structure know that, hey, who I am, you recognize me, I'm part of this mutualistic relationship. So they also build up a level of immunity to a low level of the stinging cells, okay? So that's kind of what they do there in that particular case. It's a mutualistic relationship, clownfish and sea anemones like that. But if you saw that in the movie, which a lot of you did, maybe didn't, that's what they were basically doing, was creating this kind of familiarity, so involuntarily all the synthocytes don't sting the particular clownfish and immobilize it. And then if it immobilizes it, it might turn around and eat it, considering that's like its tenant. All right, so these cells are used for defense and capturing prey. They have a little poison-filled sac, which I'll show you a picture of, and that little sac is called a nematocyst. Okay, so it's kind of like the tip of it, or the barbed tip of it, is kind of dipped in like a toxin. Works as a neurotoxin. Lots of them fire out at the same time. So basically, when the organism brushes against it, it's kind of like a mousetrap. It triggers, choo, and it's a super fast, spring-loaded little dart, microfilament type little hair that then obviously hits the prey. So this mechanism is basically uncoils like a spring and shoots out. Yeah. Basically, it's a barb. When they talk about paralyzing, it works kind of like a neurotransmitter type thing that just kind of shuts down the central nervous system to where it can't move, giving it enough time to grab it with its tentacles, and then possibly fire more of these nematocysts into it to make sure, kind of like numbing agent, draw it to its mouth, obviously, then to eat it. Okay. So basically, body plan, we jump right into, well, no, not quite yet, how they're set up. So basically, cnidarians typically have a life cycle that includes two different looks. So they have a thing called a polyp and a thing called a medusa, okay? So the polyp is the one you're most familiar with, looks like a flower, okay? So it has a tentacles, cylindrical shaped body, arm-like tentacles, and the mouth's underneath, okay? So you got your standard little jellyfish like this floating along, tentacles are up underneath, mouth, and so on, okay? So when you think of a cnidarian jellyfish, that's the body plane you think of, which they call a polyp. A medusa is just the opposite, okay? It's a bell-shaped body with the mouth on, I'm sorry, wait, I had that backwards. Sorry, Monday morning. Polyps are the upside-down trees with the tentacles like the sea anemone. Medusas are the ones that are bell-shaped. Sorry, I was totally brain cramp on that one. So, on Monday morning backwards, okay? But the medusa is the bell-shaped one, obviously with the mouth underneath, so the only thing different is the tentacles are pointed upward and the mouth is in the middle, okay? So two basically body styles. Mouth up, mouth down, tentacles up, tentacles down, okay? Both stages have a body wall, okay? So you start to see a term called gastro. Anytime you see the word gastro, they're talking about something with the digestive system. So gastrointestinal, gastrovascular, okay? So this is a tissue. Vascular refers to tissue or blood supply or, life, something of that nature. So gastrovascular cavity, okay? So I have this big basic pouch chamber, okay? A giant stomach right on the other side of the mouth, okay? Where basically all the digestion takes place. It's a slow digestional process. So in some cases, you might want to think they kind of ingest their prey alive. So it's kind of morbid but true, okay? Body wall consists of basically 3 layers. So we also start seeing, right after we go from periphera, which was an epidermis, then they went through and into the outside. Now we're actually having some regimented level of tissues, okay? And everything a lot of times runs in 3s. We know mostly for our 3 levels, we refer to as like endo, meso, and ectoderm. Well, you will see that here real soon, but the epi refers to the outside layer. Gastroderm is the cells that are covering the inner space, and then the mesoglia, before they have a true three layer, is kind of like a jelly-like substance. Helps kind of give them fill. Okay, so it's the slimy stuff you might want to say, what they call meso means middle, glia means jelly-like or fluid-like. There you go in Latin. So they do have basically a three-layer setup: epi outside, gastro inside, and then this mesoglia material inside. The middle helps give it shape. Okay. All right, so there's a simple black and white picture. I think I got a color one on here too, but showing you the layers, epi outside, showing them on both. Obviously the mouth for your polyp is here. Here's your mouth for your typical jellyfish. They draw them up and so on. All the little dotty stuff is a representation of the mesoglia, the jelly-filled stuff. So this is the actual chamber, okay? This is where actual ingestion. This whole cylindrical part is the actual gastrocavity for these polyps, okay? So this thing, and then obviously to just pushes back up here, all right, and would expand out the same way like a stomach, okay? So similar but different. Medusa, like I said, legs down, polyp, legs up, okay? There's a colored picture to show you the germ layers a little bit too as well. We like a lot of times blue on the outside, yellow on the inside, and red. It seems to always be kind of the necessary middle structure, okay? So kind of giving you an idea here. Same picture as the one before. I just added color to it so you can get a little better where the layers are, right? All right, so feeding-wise, I will start with the 7 analysis. It goes pretty quick. Obviously, they're predatorial, okay? Paralyze their play. We know they're heterotropic. We have to eat things, digest, ingest, however you want to refer to it, okay? Only has one opening, okay? Only one opening. So the digestal cavity goes in, okay? All right, and the cavity is extracellular, taking place outside the cell. All right, so basically it's no different than our own gastric biles and juices and hydrochloric acid and pepsinogen and various things, enzymes that break down our own food in our various system and our digestion tract. They just have an all-inclusive one big garbage disposal. They take the food in, starts going to work right there. Right, they ingest everything from there. All right. So basically, the digestive cavity is extracellular, meaning outside the cell. Once it's liquefied or broken down, then it's absorbed right through the cell wall. Okay? Gastroderm and digest is completed what they refer to then intercellular. No different than how we break down all of our food, and it's not truly until our own mammals that are in our small intestine where the main absorption of nutrients takes place. Okay? A lot of people don't realize that. We always think it's the stomach. Now, that's just the first staging area for stuff. Actually, your small intestine is where the majority of the absorption of key nutrients and stuff and all the various stuff is where it's mostly absorbed, okay? Undigested material then, once it's done with that, will be passed back out like in waste, the same way. But unfortunately, mouth, anus, if you want to refer to it that way, but they only have one opening, so everything that comes in, break it down, chew it up, whatever, and they just regurgitate it and it comes back out, okay? So there is waste material, okay? So pretty simple, pretty straightforward. Now, once again, respiration, circulation, excretion, 3 of them, okay? All right? So now 3 of them are all lumped into one because you don't have a lot still with the body cavity makeup. You're still simple, but you're starting to see a little bit of specialization. So diffusion, there's that infamous word, high to low over a semi-permeable membrane, lack of energy involved. So cnidarians respire, breathe, O2, CO2, and eliminate waste by diffusion through their body wall. So one of the key things too, like with those as well, that outside body wall is literally like an organ, skin type structure in which they respirate, okay? So all this takes place, breathing, circulation, obviously that, everything, the circulation is moving in and out. It's just basically diffuses straight through the body wall, okay? So obviously also with cnidarians, it's very important for it to remain moist, okay? In a aqueous or wet environment, okay? If it dries out, this process of obviously the respiration process can't take place. It begins to suffocate, okay? Response-wise, nerve system. All right, first thing ever associated, we have a thing called a nerve net, okay? A loosely organized network of nerve cells that are all that. It literally looks like a net, a fisherman's net, because under a microscope, when they look at how the nerve cells are all attached, it looks like a net, and it's all intertwined from every tentacle to every base and everything on the various structures of the cell. All right. They also have these things called statocysts, which work like an eye, but they're not a true compound eye, which we think of. But what their ability to do is detect gravity. Because think, the sun does go down and it does get dark, and if you've ever been in the water and it's pitch black and you don't have your nice, pool or lake, whatever, you go underwater, and if you can't see bubbles, which way the bubbles go up, you don't know which way's up. Well, you got some of these jellyfish that 200, 300, 400 deep. I think some of you can go past the 650-foot mark, which is, I believe, it still is, under the perfect conditions, the farthest that light can refract, meaning it gets pitch black. That's the farthest light rays can penetrate under the perfect conditions of water. Obviously, it's a lot less with the suspended matter or debris or whatever's going on. So they got to know, detect gravity. So the statisticists kind of work like that, a sense of equilibrium to know which end's up, okay? And then they have a thing called an oculi, which is not a true, it just basically detects light. So that's why a lot of times aquatic organisms, very simple, a light structure or something, a boat light, a dock lamp, or a camera light or something. Various organisms go towards that because when they associate some level of light, they detect other organisms going to that. Obviously, it could be not necessarily a socialization thing like hanging out at a party or something, but it's more like, if there's light and everything else attracted, possibly a source of food or my own speed. So there's a lot of different ways in which they process this. That's why also a lot of those bioluminescent organisms that live deep down, like in Nemo. Remember when he was getting chased around in that trench by that, I forget the name of the fish, but it had the little glowworm, caught the goggles and he's swimming around. If you haven't seen Finding Nemo, you know what I'm talking about. They use those little bioluminescent things on an antenna or something, or some level of that, to attract organisms. Oh, curious, light, attracted to it. And they're actually just using that as bait to get them in closer so they can hunt and eat, okay, because they're predatorial. So they have a little, like I said, a lot of little oculi. So here's, 3 different things: nerve net, status cyst, oculi. Okay? Here's a simple picture of what a nerve net, if you ever see what a nerve cell kind of looks like, each one of these little things is a nerve cell. There's your cell body with a dot in the middle, it's your nucleus, and then you have your dendrite types or tentacles, and they're all woven together. So they process everything in response. Okay, touch, current of water, whatever it might be. Any type of stimuli, they're going to be in tune with it. Okay, so that's why it looks like a net. Thus the name nerve net. Okay, it's a little high up there, but I get the picture. I know you have the PowerPoint there. Oops, let's turn that camera down just a smidge to get real clear. These bigger slides. Okay, so it says this is talking about movement now. Okay, so the best way to describe, it stays here. Hydroskeleton. The term hydro means water, people. Probably most of you knew that. If not, surprise, water. Okay, hydrostatic skeleton. So basically, what that means is, if you've ever had some type of container of fluid that's sealed at both ends. I know I like to use this as an example, like a slinky. Okay, if you ever played with a slinky on the stairs and it's heavier on one end and you start it, and the momentum as the metal starts to transition this way creates momentum or motions, and it flips over and so on, so you get this tumbling effect. All right. Well, imagine if it was a fluid-filled chamber that was stimulated with some electrical impulse that caused a reaction. Well, basically, that's what we're talking about, hydrostatic. So the muscles, when they contract, they're around a fluid-filled cavity, okay? Like a waterbed, okay? If you guys remember what waterbeds were, if you're sitting on one end of a waterbed and someone very large jumps on the other end of it, pushing it down, it pushes you up in the air, okay? As the water, as far as that. Or if you've seen those videos of those at the lake where you jump on those giant airbags and fling those people super 100 feet in the air, same basic concept, all right? So while the muscles contracting pushes the fluid, okay, and the medusa around, and it helps generate movement. Like basically, they slowly drift down, so when the stimuli compulse the legs, they pop, and the water that's all collected up underneath their tentacles as they swoop. It's no different than you doing like a breaststroke, okay, where you're taking your arms out and then you compress them back kind of like to your body, okay? Same basic thing is you're creating a medium, or you're pressing against a force, right? In this particular case, they're shifting the fluid in it in order to cause motion or propulsion, jet propulsion in this particular, water propulsion, okay? Pretty simple process. So they get this little bouncing action, poop, 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 okay, as they're moving, all right? Reproduction. Polyps, the upside down tree ones, remember, reproduce asexually. One of the things is at the end of each tentacle, like a lollipop, an individual just pop off, lands like a chunk somewhere on a coral reef, rock, whatever, and attaches, okay? And they grow from there. So that's one of their ways, how they asexually reproduce. They just bust a piece of it off, called budding, okay? So basically, like I said, tiny little reproducing. If they reproduce by sexual reproduction, basically everything's external in the water. They would then blast out their gametes, okay? They would release both egg and sperm. That's a lot of times where it's confusing. You assume that, okay, if they're asexual, they have both male and female hermaphroditic reproductive parts. So they're only going to, say, jettison out the male gamete, the sperm. No. In this particular case, they jettison out both of them. So you have a large colony of them doing this, okay? And they then release the egg and sperm into the water. They become externally fertilized, and then they settle like into a crevice or a rock or whatever it might be, guys and gals, okay? And then eventually from there, all right, fertilization begins. You have a term called a zygote, right? If you ever heard the term zygote, zygote is a fertilized egg, all right? Many people argue a lot of times whether it be human or Cnidarian, whatever, invert, okay? When the term zygote is referred to, it means the same thing. A fertilized egg means both gametes have met, sperm has met egg, okay? So the process of meiosis is done, okay? A lot of you guys have heard that term, even though we're not talking mitosis and meiosis. Mitosis is going on right now in your bodies. Meiosis happens one time, okay? If some of you may or may not know or may not care, here's your quick little extra tidbit on meiosis. Meiosis is the process in which the gametes meet. Haploid set, hap. Haploid means half the chromosomes, right? Diploid means means two halves, right? So if you haven't figured that out, egg has a haploid set of chromosomes or genetic material, half from mom. Haploid set of chromosomes, half genetic information from dad. Obviously, when they create the diploid set of chromosomes, right? That forms the various genetic codes. It's set, the genetic DNA for that organism, whatever it is, bird, mammal, Cnidarian, whatever, it's set. Then from that point on, okay, it goes on to mitosis, okay, which is then the cell division to one cell, zygote, goes to two to four, and so on, all right? So it takes, obviously, the two halves. So this is basically when life begins, okay? Now you're actually creating a process because you put the two halves together and let it do its thing, right? So the zygote grows. In this particular case, it has a stage. It has what they call a ciliated larva. It looks like a little tiny seahorse, okay? A little ciliated larva, okay, they call it. Has a hard surface, grows into a polyp, and then it buds into a medusa later. So they go through a set of morphosis, but as long as you understand that whether it's asexual budding or external fertilization, okay, it's still, bottom line is, okay, it goes through a stage before it becomes an adult, okay? This would be an example of a picture of budding. Like, for example, here's the base, here's the mouth, these are the tentacles. So this here is a budding process. This is almost done to attach. You actually can even see the tentacles forming here. So when this thing pops off, it'll swing down, and this base here, the skinnier base, will be the part that attaches to wherever it does. And then this will grow into a full size. This is another budding that's way far away, but eventually it'll get to this and snap off. But this is how it's just almost like a little bulge, okay? And it just kind of flexes out, turns into a mini version of it. Eventually it'll break off, settles, forms another organism. So that's just a picture of asexual reproduction, aka budding, in a hydra, okay? You don't have to know this process. I always ask somebody, do we need to know all these steps? No, but I just want to show you the life cycle from, obviously, sexual reproduction within jellyfish. In this particular case, they release out sperm and egg. They externally fertilize. You probably can't read this, but when you see your own slides, there's the term blastula, gastrula, okay? Those are terms, all the zygote, sperm, egg, gametes, blastula, gastrula. If you ever had a child development class or you had anything about human development, those are all terms you should be familiar with, okay? And like I said, it doesn't matter if it's a jellyfish or a mammal or a reptile, whatever. During the earliest stages of all life forming, they're all still the same thing, a fertilized egg, okay? That cell from one has to divide into two. Those two go to four. Eventually, when it becomes a lump of divided cells, they call it a blastula. All right, when it starts to get this kind of indentation, which I'll teach you about that later, it turns into a gastro. It's almost like taking two socks and turning them inside out when you put the pairs together. Right? So this part all right here is basically, you know, the basic blueprint of all conception between two gametes, right, or sexual reproduction, right? So and then you start to specialize, obviously, because of the cells and the genetic information that's formed. Okay. So then they go through this whole process of attaching, and it looks like little donut, looks like little onion rings here that pop off, and eventually they grow into larger ones. Okay, but just showing you the life cycle. All right, almost done here. Okay, now what you're also going to notice, and here's a hint. You're already probably tuned me out. If not, hope not. Okay, classifications of certain classes of those. I always have a handful of questions. There's none with the sponges, but you will notice from Cnidarian up, I will show you the classes of the various phylums within it. All right, and there's several classes here. I always ask a simple question from it. These are the ones everybody always misses in multiple choice questions, because you study the 7 essential plus the characteristics of them, but you forget to look at the different groups in them. So don't forget those last couple slides that are associated with the quiz for that day to look them over. That's just a little extra hint. I always do ask, you know, information. So the first one says a zoa there with an H, and you got hydrozoa. Basically, these are all your jellyfish. This name refers to the cup. So all of them that are cup-based, your medusas. Okay, the largest one, 4 meters. I think that's the lion's mane one. I think the one you've probably seen pictures of. They're massive, 4 meters across, a meter thick. So take 4 meters across that. So that's a pretty good size chunk of a jellyfish. You got your hydrozoas. One you're probably familiar with, Portuguese man of war. Okay, and things of that nature. The polyps forms. Most hydrozoas have branching colonies as well. Okay. One thing a lot of people don't realize with hydrozoas is they're actually a colony of a multitude of polyps. Polyps meaning that type of. Everybody thinks a Portuguese man of war is just one giant organism. No, it's actually a bunch. Okay, polyps within a colony that have certain looks to them. Right, so it looks like one general thing, but the actual tentacles and all the various things, they actually form different structures. So the actual balloon part you see at the top is a separate type of polyp, and the tentacles are separate polyps, and then even polyps that digest the food and so on. So it's just an interesting thing about hydrozoas. Okay, no big deal. You can find them in freshwater. The ones in freshwater don't have a medusa stage. It exists only as a solitary polyp, meaning as an upside-down tree. They can reproduce asexually or by budding, as you know, and they can even produce by egg and sperm or sexual reproduction. Okay. So remember, when they talk asexually, it's usually going to be some separate, smaller, faster, quicker process. But anytime they refer to gametes and sexual reproduction, they're always going to refer to the same basic concept. There's got to be 2 gametes. There's got to be 2 haploid sets of genetic material to form a diploid set to get a zygote. Okay, it doesn't matter the organism. Anthrozoa is all your sea anemones and your corals. Okay, so there's 3 classes. I think there's only the 3 classes of them. That's right, yeah. All right, and some characteristics for that class. I think I came up with 6. Anthozoas can also undergo budding and procedure sperm. So I basically give you some characteristics. Sea anemones are also solitary structures. They sting and catch. That was what Marlin and Finding Nemo lived in, was a sea anemone with the tentacles upside down. Okay, so it means it looks like a flower with the tentacles upside down. Flower. All right. There's some pretty ones there, some examples of some true polyp ones. True medusas of this hydra. Okay, as long as these tentacles are, this is a deep water one that actually has a bioluminescent type, really pretty. I forget the name of them. I think that's the lion's mane one. Here are some hydrozoa ones. There's your typical Portuguese man of war. So what they were saying was, this is a separate polyp. The tentacles are a separate polyp, and so on. Okay, so all different types. And there's your typical sea anemone, like Finding Nemo. A funny thing too, you might see in some of the sea videos, a lot of times these sea anemones will be kind of coerced to be attached to the back of lobsters for protection. So they hitch a ride and it looks like this thing has this giant, crazy, despicable me, wild hairdo little minion on its back, and they can have as many as 2 or 3 of them stuck on its back, right? Because every one of these cenocytes have all those little tiny tentacles. Every one of these are full of little poison sacs that then they would grab, draw in, and eat. Okay, There's a picture of coral. The red stuff is fire coral. If you guys ever go scuba diving, don't touch it. Okay? Fire coral is just what it says. Makes your skin feel like it's on fire. Okay? If you brush up against it, they have all them full little tentacles too as well. Okay? So if you ever go snorkeling, you ever go noodling, lobstering up and down the Florida coast on the east side, don't bump into that stuff, or a lot of times just wear a long sleeve t-shirt. That's what I learned how to do. Is I actually brushed up with my calf doing that once. Ooh, it was just annoying. All right, it was like almost like I would call it like same thing as road rash. Like if you ever slid, you got a strawberry or something, slid in the grass. It kind of was like that, just annoying as heck, burned all day long. All right, they have a lot of bacteria. So yeah, if you ever see that red stuff, avoid it. Okay. And last but not least, ecology of all corals. Hard or stony corals that build reefs require high levels of light because they rely on symbiosis. They're protected, divers, pollution, silt, so the little tiny fine particles kill coral reefs. They clog it. They clog the holes. Okay. So when you see in Hawaii, if you've ever been to Hawaii, you see a lot of it. You don't really see much of it in Florida, maybe down farther down in the Keys and so on. But there's always busted pieces of white coral that's washed up on the beach. That's dead coral. Okay, it's been bleached out for whatever reason. All right, when you see it like that, it's dead. Okay, but they call it chlorobleaching, which is not good. The coral loses its color, becomes white, causing a problem. Coral reefs are rivaled only by the rainforest. We know the importance of this now. You don't hear of it diverse as much. They are extremely protected. Not only that, they used to take a lot of, and they may still do, decommissioned warships, take any things that are biohazardous off of them after we destroyed so many coral reefs due to World War II and various other commercial activities. They would blow them up, sink them, and create these natural reefs. So when you're flying in a plane and you look out the window over the ocean, you see all those cool different directions of the white water breaking. When you see it start breaking, you know there's some type of continental shelf or some type of obstruction underneath that causing that. Possibly a lot of times, usually it's some type of a coral reef or rock outcrop. Okay. So a good example of that would be if you ever seen the movie Castaway with Tom Hanks, where he goes up on top of the little island he's stuck on and he looks out, and it shows all the cool different directions the waves break onto the island, and you can see how the different things, that gives you an idea how the lay of underneath the water is all the coral reefs are. Okay. All right. And last picture, these are the little tiny discs. So basically, if you were to brush up against this little microfilament hair, this spring-loaded little dart is what comes blasting out and zaps you with this little stinger. Okay? And that's what gets you, right? So these are the little undischarged, and then they got a recharge. So like they pop like little one-time things. Okay? I think in the video on too, it shows two sea anemones like attacking like a time lapse, but you see all the little tentacles coming out and attacking each other. Okay? And that's it. Okay. So let me go here to light back. Wait, we'll turn the light back off real quick. Hold on one second. Let's go in, almost done here. Back to 3. On my glasses. It's okay. So all I have for you here for right now is this one right here. It's a little simple video, 10-15 minute type one with about 13 little guide there to it. Right, just a little extra visual part of it, something very simple with that, okay? So that's the only new stuff right now I have for you associated with Cnidarian. I have a web quest, and I'm having some struggle with it, but I'll let you know if I get that. It was a combo with sponges and Cnidarian, but I can issue that on Thursday, the same day of the quiz, so I'm not really worried about it right now. Okay, so what to do of you real quick. Okay, so couple things, and then I'll let you guys get. All right, number one, priority like that is on Thursday. Everybody that's logged in here, let's see, I got looks like 16 of you here. So, oh, good, all of you here. So you have a short quiz. Okay, I'm going to show you the slides real quick. So the slides, all right, real quick view from present. We always forget on this first quiz. I have, okay, so from slide 1, your Kingdom Animalia, okay? All of these, I take some simple questions, so all the slides that I went over to that sponge, to the end of today's. So from the first slide to the slide that says Nigerian we start today, this is not on. So nothing on the quiz Thursday over Nigerian. Only from the lecture and from last Thursday. So if you need to know the exact slides, exit that. So that is slide basically 1 to 17. If you needed to know specifically, a lot of them are pictures, of course, but it's not as much information. So if you looked at the slides on the side, slides 1 to 17, and I'll put that in a little reminder. I said, do not forget to look over slides 1 through 17 for the quiz over AK, Intro to Animal Kingdom, and Phylum Porifera. And that's it for the first quiz. Okay? So it'll be, I think, and I usually divide them up. Probably what I will do is I usually divide them up into two, meaning the first quiz would say Kingdom Animalia, second quiz would be Porifera. It's just how I was typing them up because I have to put them in since you guys are now online at home doing those. Unless you wanted to take it here in person, you're more than welcome. All you gotta do is pop in sometime that day. You can do a written test right in here with me if you want to as well. Okay, so that is Thursday. Quiz on Thursday over the first 17 slides from Thursday. Ooh, that's funny. Thursday. Okay, all right. So, only thing you have for is this little simple reading excerpt, short little video right here, okay, by Friday. Should take you 5-10 minutes if you want to do it now or not when I release you, okay? Second, I also want to give you time is make some time. Think about this week, getting in here and working on your semester project. If you haven't yet, make time to do that. Remember, I am here 7:30 to 3 every day, Monday through Friday. Wednesdays is a study hall day. Doesn't matter though. I had lots of people still come up. I even had lots of people last Friday during their 6-hour class just came up here live while I was lecturing, and they sat in class and worked on their project at the same time while I was going over the material. So I don't want excuses as to not have it done come by the 22nd of December. Merry Christmas. Okay, there we go. All right, so other than that, we're good. So we got about 20 till 12. Which is a lot, 20 till 12, 20 till 9, okay? So I'm good to go for today, all right? So does anybody have any questions on anything? I got all of you here. I'm looking at all your shining faces one way, shape, or form, all right? Some of you, looks like some of you look like you're in some classrooms throughout the building, so I'm here if you needed to come down. Also hard copies. If you needed some hard copy that you had problems with, whatever, come by and get them. This one's only a little 13 little thing, as you see there, a little reading guide I created from watching the videos. So most of you did that on your own notebook paper or do it on that and take a picture, doesn't matter. So if you needed a hard copy of that, great, I got them. Most of you probably don't want something like that. Okay? So any questions, comments, concerns, problems? No? 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 Okay. All right. All right, so if that's the case, then without further ado, like I said, I'm good to go. So I'm going to stay here. Since I had you guys log in there, it took us about 40 minutes covering all that stuff. I'll do a little recap. I'll do a little entry either on update or just I might send up, hey, don't forget to look at the little bullet points just for a reminder. A lot of you guys are probably, hopefully, if you haven't, are taking notes in the sense of get one of those little $2 little flip little 2-inch by 4-inch pads and say, all right, what I got to do for Bio 2? Myron reminded me, got this little thing to watch, 13 video, quiz on 30, something of that. If you got a whiteboard or something, make a mental note of it, okay? All right? So that's all I got for you for today, okay? So I am done with you for 45 minutes. So you have the rest of the time to finish that little reading guide or whatever you need to do, okay? Remember, semester project, 7:30 to 3, come up here whenever you want, okay? All right, you guys are good, man. Have a good rest of your day. I'm good. Okay, I'll stay here if you need something. I got my mic on. Miss Washington, you got a question, dear? No? Okay, cool. You're good. I'm right here if you need anything. Okay, or not.